Good morning, good afternoon, and good evening. I'm Wafa El-Sadr. I'm the ICAP Global Director, and it gives me great pleasure to welcome you today to this webinar entitled SARS-CoV-2 Serologic Testing. I'd like to invite Mark Harrington to also say a few words of welcome. This webinar is co-sponsored by ICAP at Columbia University, the Treatment Action Group, and the COVID-19 Working Group. Mark?
Oh, thanks, Wafa, and good morning to everybody. It's exciting to see 250 participants. Today, we're going to hear from some really excellent virologists and immunologists about some of the new SARS-CoV-2 antibody tests and what are they and what do they mean? And the answers are going to be incredibly important to enable our city, our state, our nation, and people around the world to begin to come out of isolation and to resume social and economic activities to recover from this terrible pandemic. We will be taking questions, and if you see in the bottom of your screen a chat box, please, a Q&A box, please type in the questions and they'll be transmitted to our excellent facilitator, Jessica Justman. We have a lot of really exciting presentations, and so without further ado, I'd like to move on to the panel. Thanks, everybody, again.
Thank you, Mark. And I'd like to just introduce very briefly Jessica Justman, who will serve as the moderator for today's webinar. And Dr. Justman is the Senior Technical Director at ICAP at Columbia University, and she's also Associate Professor of Epidemiology at the Columbia Mailman School of Public Health. Jessica?
Great, thank you very much, Wafa, and thank you, Mark, and good morning and good afternoon to all of our colleagues who are joining us today to talk about serologic testing, what it is and what does it mean. I think we all know there are many different tests. There seem to be more tests out there every day, and there are many unanswered questions about these tests. So today we're going to review basic information on how the tests work and how to interpret them. And we're going to look at this through the lens of recent work done here in New York City at Mount Sinai. And then we'll also look from the perspective of the New York City Department of Health and from the perspective of the New York State Department of Health labs. And then we will widen our lens and think about this whole topic even more broadly from the perspective of modeling the pandemic at the population level and what the testing will and will not allow us to do in understanding what's going on at the population level. So let me begin by introducing our first speaker. Dr. Florian Kramer is a professor of vaccinology at the Department of Microbiology at the Icahn School of Medicine at Mount Sinai. Most of his work has focused on influenza, including immune responses to influenza and other RNA viruses, with the goal of developing better vaccines and novel therapeutics. He holds a PhD in biotechnology from the University of Natural Resources and Life Sciences in Vienna. So Dr. Kramer will begin, and I think just to, again, underscore what Mark said, people can put their questions into the Q&A box, and we'll take all the questions at the end. Okay, now we can please go ahead and move forward with Dr. Kramer's talk. Thank you.
Thanks for this nice introduction, Jessica. I'm just going to share my slides here. Let's see if this works. I think it does. I hope you can see my slides. And so I'm just going to give a brief intro about what SARS coronavirus 2 serology is and why we need it. And then I'll show a few things that we did so far at Mount Sinai. But I wanted to start with very much with the basics. So the question is, what serology? So when we get infected with a virus or any other pathogenic bacteria, basically parasites. Our B cells make antibodies to this invading microbe, and these antibodies in many scenarios neutralize or kill the microbe. And so these antibodies can be found in the blood serum, and that's why we call it serology. And I just wanted to show you a graph here on this slide about when serology makes sense. And this is not specific here for SARS coronavirus 2, but typically when you get infected, you develop symptoms. If this is a virus, the virus starts to replicate. You find viral RNA and viral antigen in your body. And this is when you typically do nucleic acid-based or PCR testing. And that's nothing else for SARS coronavirus 2. That's when you get tested for viral genome. And serology testing usually comes up a little bit later. There's an overlap of IgM antibodies, which is a type of antibody that might already be there while you're still in the acute phase. And then typically IgG comes a little bit later, and that's when serology testing usually makes sense. In most cases, specifically for SARS coronavirus 2, serology is not very helpful to diagnose acute infections. And that's something you have to keep in mind when you look at nucleic acid and serology testing. So why do we think is serology useful for SARS-CoV-2? First, and that was something that we did initially, was we wanted to screen for donors for convalescent plasma therapy to treat severe COVID-19 cases. And for that, you need to find people who have high titers of antibodies. It can also be very useful to afterwards confirm that somebody was in the past infected with SARS coronavirus 2. It's very useful for serosurveys to determine the true infection rate with a pathogen in the population. And we're hearing a lot about asymptomatic cases now. Serology might be a way to figure out how many there are. It's a very important tool to understand dynamics of the antibody response. And in the long run, we can establish correlates of protection. We can figure out if somebody is protected by the antibody response. And we can try to figure out how much antibody is actually needed to protect somebody and how long that protection lasts. Now, if we look at coronavirus, you have a model here on the left side. In the virus, or on the virus, we have several proteins. Inside we have the genome, which is covered by the nucleoprotein. Then we have the viral envelope with the viral envelope is the envelope protein and the matrix protein. And then we have this large spike protein on the outside. And so we chose to use that spike protein as target for serology. Other people have used the nuclear protein with a lot of success too. The reason why we used the spike protein was because it is the target of neutralizing antibodies. And you can see the spike protein here on the right side. Most of the neutralizing antibodies actually target this receptor binding domain, which is on this spike protein. And that's the part of the spike that interacts with the receptor that the virus uses to get into our cells. So for us, that was a good target for serology. And what you can do then is you need to get these antigens made, these spike proteins made, and you can do that by recombinantly expressing them. And in our lab, we did that by making the soluble version of this spike protein, but also a soluble version of that much smaller receptor binding domain, which might be useful in some cases. And so I'm just going to show you how typically these assays work on a more general basis. You make these recombinant antigens, and they are usually immobilized on a surface. Could be an ELISA plate, could be any other surface. And then you take your serum from your patients or the individuals that you want to look at, and this serum has antibodies. And here we have the case for negative serum. So you put your antibodies on this coated ELISA plate, for example. And what happens is if it's negative serum, the antibodies just don't do anything and they don't bind to your antigen. Now, if you have a positive serum with specific antibodies, these antibodies actually stick to the antigen. And then in many cases, you add a secondary antibody that now recognizes these human antibodies. You add that, and that sticks to the human antibody, and then you get a signal, and you can measure that signal. There's different variations of how these assays are done, but that's kind of the basics. And so that is what we do with serology assays. And then there's different types of tests, and that's also something you have to keep in mind. So in general, and these are just examples here, there's two types of assays. One assay that gives you either a yes or no response. And these are, for example, lateral flow assays that a lot of you are familiar with, where you just basically have a line that shows up, a control line that tells you that you did the test right, and then you see if the result is positive, if there is a second line or not. So these tests just give you a yes or no answer. And they can be linked or loosely associated to protection. They cannot be used to figure out how long you will have antibodies because they don't give you the level of antibody, but they're easy to use, and you can use them in large numbers. And they can even be used as point of care test. And then you have more quantitative assays, and an example is here in ELISA, but there's many other assays that can be done in a quantitative way. And what they give you is actually a titer. So you basically learn how much antibody there is in the serum of a person. And so this quantitative data can very firmly be linked to protection. And we have done that for influenza, we have done that for measles, for many other examples where we know how much antibody you need to have to be protected from infection. And this can, if you have a very good idea about the dynamics of the antibody responses, also be used to predict the protective period. The scalability is a little bit lower, and usually these assays are only performed by specialized laboratories. Another point that I wanted to bring up is sensitivity and specificity, and that's very important. Those are the criteria that I use to look how well assays work. Sensitivity tells you how many real positives are detected. And for serology, that is very often influenced by when the sample is taken after onset of symptoms. And the specificity is basically telling you how many false positives you're detecting. And both of these should be in the very high 90% range if possible. And the other recommendation that I have is you should only use tests with known performances. And if possible, they should have at least an FDA emergency use authorization. So they should have checked metrics. So another thing to keep in mind is that different tests are useful for different things, right? If you want to do a large serial survey, you know, lateral flow tests might be more ideal. If you want to do a study to look if what the protective titer is, you need something that's more quantitative. And so just to show you what we did here at Mount Sinai, here we developed an enzyme-linked immunosorbent assay, or ELISA, and these are some of the results that we produced here. And basically what you can see here in these curves, these are dilution curves. We had negative controls in black and green, which did not bind to the RBD that we produced or the spike, but samples from people who had infections with SARS coronavirus 2 did bind very well. And you can also quantitate that, and you can appreciate that there is actually very nice difference between the control samples and the seroconverters. And what we also did is we looked if these ELISA titers, which are easy to generate, correlate with neutralization. And neutralization is much harder to test because you need a biosafety level 3 laboratory for that. And so we did that, and we saw that what we produce in the ELISA assay actually correlates very well with neutralizing activity, which is good. And so far at Mount Sinai, we have screened more than 22,000 donors, plasma donors by ELISA, and they are mostly mild cases, and they come in two different types: people who are PCR confirmed donors, and people who are donors with suspected COVID-19 infection, but they have never been PCR confirmed. And so far, we have treated more than 250 patients with convalescent plasma. We just put a preprint out that shows the result for the first about 1,400 donors in terms of ELISA and also PCR positivity. And we found something very interesting here. On the left side, you can see a pie chart of PCR confirmed donors. And we found that 99.5% of those PCR confirmed donors actually made antibodies at some point. And only 0.5% were negative for antibodies. So that tells us that basically everybody makes antibodies. What was also surprising was that the COVID-19 suspected donors, we only found about 40% of them had antibodies and about 60% of them did not make antibodies. And those are people who think they had COVID-19 and had symptoms that were consistent, but it was never proven that they actually had it. And that kind of tells us that there's a lot of people out there who assume they had it, but might not actually have had it, might have had other infections. And this is just a closer look at this data. On the left side, you see the different titers that we measured from negative to very high titers. And initially we had people who were PCR confirmed and were negative, but then we recalled them usually 1 to 2 weeks later. And basically all but 3 of them then had titers and had an increase in ELISA titers. And so just to conclude, what does this all mean? Usually antibodies are a good thing. Antibodies protect us from reinfection for most viruses that are out there. And neutralizing antibodies are usually associated with protection. So measuring neutralization is important, or having an assay that correlates with neutralization is important. And we also know that antibodies protect us from infections with human coronaviruses like 229E or HKU1 or OC43. So we can make some assumptions for if they're protective against SARS coronavirus 2, but we still need to prove that there's actually a protective effect. And we also need to figure out what titer is protective, and this is very important for vaccine development. And we have now started studies where we follow people that already have antibodies for a year to see if they can get reinfected, if there are breakthrough infections, and at what titer these breakthrough infections occur, to actually figure out what titer is needed to protect you for a longer time. And with that, I just want to thank my team and my collaborators, and I wanted to show this funny picture that somebody sent to me. So thanks a lot.
Great. Thank you very much, Dr. Kramer. That's a great last slide for us. So now we're going to move to our second speaker, Dr. Jennifer Rickman. Dr. Rickman is the Assistant Commissioner and Laboratory Director for the New York City Public Health Lab, which is part of the New York City Department of Health and Mental Hygiene. In this position, she's responsible for overseeing citywide testing during infectious disease outbreaks, such as Ebola, Legionnaire's disease, Zika, and now COVID-19. She holds a PhD in microbiology from the University of Washington. Dr. Rickman.
Thank you so much, and good morning and good afternoon. So as Dr. Justman said, I'm Jen Rickman, and I'm going to talk to you about testing for COVID-19 from RNA and antigens to antibodies. Let's see. Sorry, my slides are not advancing. There we go. So just a little bit about where I work and what we do here. The New York City Public Health Laboratory is part of the New York City Department of Health. Our testing here is focused on the health of the population of New York City. We like to say that we save lives, 8.5 million at a time. And the testing that we do here, the work that we do in the lab, focuses on outbreak detection and response. Much of what we do is focused on infectious disease outbreaks. We also work on biothreat agents, and we do a fair amount of environmental testing as well. And right now we have about 200 staff that work in the laboratory. We're located in Manhattan, and there's a lovely picture of our lab there. So to back up a little bit, I want to talk first a little bit about the different types of testing that are available for coronavirus. The first being the tests that detect viral nucleic acid. These generally are nucleic acid amplification tests, or NATs. They detect the viral genetic material or the SARS coronavirus 2 RNA in a patient specimen. These tests are best for diagnosing acute infection, and they're useful for informing clinical evaluation and in a hospital setting, for example, directing infection prevention and control practices, or for a physician to, for example, tell a patient to stay home until they get better if they can manage their illness at home. Generally, these tests are performed on respiratory specimens, specimens like nasopharyngeal swabs or NP swabs, throat swabs, nasal swabs, and now even saliva, as well as lower respiratory specimens like sputum. The next type of test, and this is very new on the market right now, are tests that detect viral antigens. So these tests detect proteins from the virus in patient specimens. Again, they're useful for diagnosis of acute infection because they're detecting the virus. They may be a little bit less sensitive than a NAT-based test and are performed on respiratory specimens like nasal swabs. And then the third type of test, which is what Dr. Kramer was just discussing, are the tests that detect the immune response to the virus, antibody or serology tests. And again, these detect antibodies specific to the virus that are present a few weeks after infection. They are not useful for diagnosing acute infection, but do give us evidence of a previous infection and generally are performed on blood. So let's dive a little more deeply into NAT tests. These were the first tests that were developed and available for diagnostics for SARS coronavirus 2, and they're based on detecting the sequence of the viral genome specifically in a patient specimen. And they do that, we do that by amplifying those specific nucleic acids and then detecting the amplification of that target. These tests can be manual or automated and be kind of at a low throughput or a high throughput available. So this picture here is of an ABI PCR instrument that we use for more low throughput or manual tests. And then this instrument here is a large Cepheid instrument. It's actually the size of a big deli refrigerator. And basically this guy has about 48 PCR machines inside it. And each of these little cartridges can go through and test one patient specimen. The Cepheid test takes about 45 minutes to do. Testing using a more manual extraction and PCR method can take 6 or 8 hours to do. So when we do these nucleic acid amplification-based tests, there's 3 main steps, and we've run into some issues with all 3 as we've gone through this outbreak in the US. The first step is to collect the specimen from the patient, and we've seen shortages of collection kits, and you may have heard about all of this in the news, things like swabs and viral transport media, which is pictured here. It's the juice that we put the swab in to get the swab from the patient to the lab to do the testing. The next step, once we have the specimen, is to do an RNA extraction. This gets the nucleic acid from the specimen, which may include viral nucleic acid, out from the cells in the specimen, that then we can do the third step, which is amplification, where we do a PCR-based test, for example, to amplify viral-specific targets to determine whether or not the virus is present. So what we've done to address these issues, when we look at collection kits, we've started to look at alternative devices. So things like 3D printed swabs, which are pictured here, using an alternative to the stuff in the pink stuff in the tube I just showed you, like saline. We look to use alternate specimen types. So many tests were initially only available to be run on things like a nasopharyngeal swab, which is a swab that goes up your nose all the way to the back of your throat. We've now started to validate specimen types like a nasal swab, so something more like a Q-tip just going up your nose, and saliva to look for the virus. To address RNA extraction issues, we've looked at alternate platforms for doing this step, as well as methods that allow us to kind of skip this RNA extraction step altogether, and there are some platforms that are able to do that. And then amplification, initially there were a lot of shortages of the PCR reagents, and those have largely been mitigated by increased availability of those reagents. So a new technology has recently come on the market using the CRISPR type system to detect SARS coronavirus sequences in patient specimens. This test is called the Sherlock CRISPR SARS coronavirus 2 test. It was developed by groups at the Broad and MIT and Harvard. This test is performed on an upper respiratory swab specimen. So you collect the specimen, you extract the RNA from the specimen and amplify the target in a method called reverse transcriptase loop mediated amplification. It's sort of like PCR, but a little bit different. And then the detection step uses the Cas system, where a guide RNA detects and interacts with the viral target in the specimen, and this initiates cleavage of a reporter, which results in something that we can detect to determine whether or not the test is positive or negative. So this is really sort of cool from a very lab nerdy standpoint, because this is a technology that has not been used for viral diagnostics before. This test just came out very recently. So moving along to antigen tests, there's one test on the market. It's made by a company called Quidel. It's the Sophia 2 SARS antigen fluorescence immunoassay, and it detects the nucleocapsid protein from the virus. So that's this protein here shown in red, which is inside the viral capsid. So a specimen is put into a specific reaction tube, and in that tube are reagents that disrupt the virus and expose this protein. And then the specimen is applied to a test cartridge, which looks a little bit like the cartridge that Dr. Kramer just showed you in his talk, where the protein is captured by specific antibody on the in the system, and then that protein is detected in a reader. This test is authorized to be performed at point of care. It has a slightly lower sensitivity than a nucleic acid amplification-based test, but it's very quick. It can be done in 15 minutes and can be done in a doctor's office, for example, to detect whether a person might be infected with the virus. Then I'm going to move on and talk a little bit more about serologic tests. So these are the lateral flow-based tests and the more lab-based tests that Dr. Kramer just described. Again, these are tests that are intended to detect antibodies to the virus, and they can be specific for a certain type of antibody, like IgG, which comes up later, and that is the type of antibody that may provide protection, or IgM, which comes up earlier in infection, or a combination thereof. A positive result indicates that the presence of antibodies likely resulted from an infection with the virus. There are some tests out there that may have some cross-reactivity with related coronaviruses, so that must be taken into account when somebody's choosing a test to use. A negative result can be interpreted as no previous infection, or that the patient was tested too soon before antibodies were able to develop. So as discussed, at this time, we don't know whether antibodies provide protection against reinfection with the virus. So right now, if a patient comes in and has a positive test, we can't use that result to say, you can go to work, you can not wear your mask, you can relax precautions. We don't know that yet. And so a positive test should not be used to change anybody's behavior. We can use this now, and serology is very important for determining prevalence of coronavirus infection among a population. This is really important from a public health standpoint. We want to know how many people have been infected, where the virus has been in New York City, for example. And as Dr. Kramer said, it's very useful for identifying patients who may be candidates to donate plasma for therapy. So a little bit about how tests are used in the US. During a public health emergency, the FDA can authorize a test through the emergency use authorization process, which allows tests to get approved very quickly. CLIA is the group that categorizes tests after the FDA grants authorization. And so CLIA can categorize tests as high complexity that need to be performed in a laboratory, moderate complexity, which still need to be performed in a lab, or waived. And these are the types of tests that can be performed point of care. These tests are simple to perform and have a lower risk for producing errors. They're not error-free and need to be done carefully, but they are less likely to produce an error, easy to perform, and so therefore can be done at a point of care setting. Tests that don't have an EUA default to being categorized as high complexity and must be performed in a laboratory. So there are a number of serology tests that have this emergency use authorization. There are two that may be performed only in the lab with the EUA. One you'll hear about next, that was developed at Wadsworth. one from Mount Sinai that you just heard a little bit about. And there are about 10 kits commercially available that have EUAs, and all of them must be performed in a laboratory setting. So there are no serology tests right now that can be performed in a point of care setting. The FDA has this actually very cool performance data and negative predictive and positive predictive values, sort of gives you an idea of the sensitivity and specificity of a test. And I left the link here that you can kind of look at that later. So some of the important performance characteristics that Dr. Kramer talked about, you can get a little feel for how those work out in terms of testing big populations and what that means. So recently, the FDA had a policy where tests that were not validated or reviewed by the FDA could actually be put on the market. And there was an issue that manufacturers were falsely representing their tests as a FDA approved and marketing them as point of care tests. And there were some places where these tests that were being used inappropriately at the point of care, which is dangerous because the performance of these tests was unreviewed, and they may have been producing false results and giving people a false sense of security about their results. So there was a new policy change based on some data that had been coming out, and I have a link here to some of that data showing that some of these tests were not actually performing very well at all. And so now the FDA has changed their policy a bit, and all test manufacturers do need to submit data to the FDA for an EUA within 10 business days of notification. And so the tests that had previously been listed on the FDA website now need to go through this process by this coming Monday or pull their tests from the market. The FDA has given now specific guidelines on performance thresholds for sensitivity and specificity of serology tests and developed a streamlined process for submitting tests to the FDA for EUA authorization. So in the coming months, how will we use these tests from a public health perspective? So one, viral RNA and viral antigen testing will be used for diagnosis of cases. And those diagnoses will be followed up with contact tracing that will be used to curb the spread of SARS coronavirus as we move forward in our where we are in the outbreak and try to lift some of our mitigation policies. Serology data will be really important, and serosurvey data will be used to detect past cases and determine how widespread coronavirus was in New York City, and again, provide insight into the immune response to the virus. So that will be very important for us from a public health perspective. So I've included just some resources where you can find more information about what's happening in the New York City Health Department around coronavirus as well as nationally. And I will end with this view from inside our lab. Thank you.
That's really great, Jen. Thank you very much. Another terrific talk and a terrific ending slide. Our next speaker is Dr. Jill Taylor, who's the director of the Wadsworth Center, New York State's Public Health Reference Laboratory. Dr. Taylor has worked extensively at the federal level and has served as a member of the National Library of Medicine Board of Regents and the CDC Board of Scientific Counselors for the Office of Infectious Diseases. She received her PhD in microbiology from the University of Queensland in Australia. And I think now we can turn this over to Dr. Taylor. Thank you.
Good morning, good afternoon, good evening. Thank you very much for the invitation to talk at this webinar and to share our knowledge and our questions about this pandemic. Just a little bit of background about the Wadsworth Center. We are New York State's Public Health Lab. Jen talked about the New York City Public Health Lab, and we always have an inside joke that New York City really is its state on its own, and so that's totally fine. We have two big labs in New York State. Our role really is as a reference lab for both surveillance and outbreak investigation. We also have a pure research arm in our lab, and we have a regulatory arm. So we wear multiple hats. Next slide, please. I'm going to jump straight into our role in the serology testing. Clearly, when we became aware of this pandemic, there was a huge ramp up into molecular testing. That continues. We're testing of the order of 1,000, 1,500 specimens every day for molecular testing, and now we're ramping up our serology testing in several areas. It's important to remember that our role really is for public health purposes, and so we design tests that are going to answer the questions that are needed to guide the public health response. We've been working closely in the area of qualification of potential plasma donors with the New York Blood Center, with Westchester County, and with Florian's group at Mount Sinai in this area, and design tests for that purpose. Clearly, it's important to ensure the safety of our first responders. And while at the moment we don't know, as both Jen and Florian have said, that because you have antibody, it means you're immune, we don't know the answer to that question, but we are able to test our first responders, and the plan is to monitor them over time. Sero surveys are going to be incredibly important in the future as we monitor the evolution of the pandemic, and so we're doing a lot of that. And from the sort of research perspective, we're a science-driven organization, and so it's important to understand the immune response to this new virus. So I'm going to jump right in now on the work that we're doing. Next slide, please. So we have 3 tests at the moment that we're using. The first one, as Jen mentioned, is FDA EUA approved, and we look at that as a first-generation assay. It's antibody microsphere immunoassay, which works in many ways like the ELISA assay that Florian described, except that instead of a flat ELISA plate surface, we bind the antigen to a bead, and the advantage of the microsphere assay is that you can multiplex it and have beads bound to multiple different antigens so that you can simultaneously look at an antibody response to different antigens. At the moment, the one that's been FDA EUA approved actually uses the CoV-1 N antigen, and I'll tell you why in a minute. We are working on a second generation with the CoV-2 N antigen and the RBD, and that'll be submitted to the FDA for approval very shortly. We also have, as I said, a regulatory program in New York, and we are able to do tests under New York State's Clinical Laboratory Evaluation Program approval system. And we have an MIA using blood spots as a sample source. Our EUA approved assay uses venipuncture blood, but we doing high volumes for us with blood samples is logistically difficult with all the accessioning and processing you have to do. And so we have adapted blood spots as a sample source. And in that case, we're using the CoV-2 N antigen, and we'll also be using the spike antigen, and we'll be submitting that for EUA approval shortly as well. And we have a plaque reduction neutralization assay, the classic old virology plaque reduction test that we learned as junior technicians and postdocs. It uses the BEI Washington 1 strain of SARS-CoV-2 in Vero E6 cells. Next slide, please. So why on earth did we use the CoV-1 N protein? I'm sure you're all thinking. And the reality was that we had it. We had developed this antigen in the SARS outbreak of 2003, and we had a very good source of it. And so we started using that, understanding that the similarity between the N protein for SARS-CoV-1 and SARS-CoV-2 was very, very high. And we now have developed the CoV-2N, and you can see that they behave very, very closely together. We're now switching over to the CoV-2, but the CoV-1 has been very useful to us in the early stages of the development of these assays. Next slide, please. So when you're thinking about a serological assay, there's two really important things to consider: specificity and sensitivity. And Florian did talk about these, as did Jen. I want to show you in this figure, the red dots on the left are sera collected from PCR positive confirmed SARS-CoV-2 patients, and the blue triangles and squares are sera from people, one infected with NL63, one of the commonly circulating coronaviruses, one from flu, one from metapneumo, and the other from other respiratory pathogens. And that you can see that there is definitely cross-reactivity with sera from those patients to the N protein. Now, this is important to know, and it's especially important when you're looking at the laminar flow assays that Jen showed, because you can't quantitate a laminar flow assay, you've really got to understand that a positive is a real positive and not a cross-reactivity. And so we've made our test quite rigid, if you like, and used 6 standard deviations above the baseline to require positive a result of positivity. If you go to the, and there's some discussion about whether we've made it too rigid, but that's what we're using at the moment, because we want to be sure that if we say there's antibody, it really is antibody specific to the COVID-19. If you go to the bar of dots on the left, you can see a whole lot of dots right down the bottom. And these were all collected less than 10 days after symptom onset. And so clearly in our tests, because of our 6 standard deviations, we're losing low positives, and we're aware of that. But we think that it's for both qualifying plasma donors, potential plasma donors, and also qualifying antibody that we hope is indicative of immunity, though we don't know that yet. It's important that we result the strong positives. So these are just 2 things that you need to be aware of. And sensitivity clearly depends on timing of collection, as Florian said. Next slide, please. So in this slide, I want to show you the relationship between the antibody levels and the neutralizing activity in our assay. And again, it's this question of stringency. You can see that the red dots show use of the CoV-2 RBD protein, and the blue dots show the use of the CoV-2N protein in the MIA. And you can see in both cases, we're showing here the PRNT50, which is a 50% reduction in plaque number on the left, and the PRNT90, which is a 90% reduction in plaque number on the right. And you can see at a 50% reduction, there are many people who have good neutralizing titers up to about 320, which we consider sort of 320, 160, 320 is about as low as we think it is indicative of immunity, though we're not sure of that. And so you can see at the 320 in both the RBD and the N, there are many people that have antibody at this level. If you go to the 90% reduction 2 graphs on the right, though, you can see, well, that would be your left. You can see that the majority of the individuals are at less than 1 in 80. So this is an important question that we have to think about going forward. What level of both MIA antibody and neutralizing antibody is important for immunity. And we don't know that, but we want to have done the analysis so that when studies of protection after plasma donation or after reinfection become available, we know what sort of antibody levels they had. Next slide, please. And this is just to show you the timing of development of the antibody response. This is in donors who have a titer, an MIA titer, a neutralizing titer of at least 160. And you can see that the titer reaches a peak around about 30 to 35 days. Earlier than that, it's considerably lower levels. We're a little bit worried about the rapid drop-off after 40 days. We don't know whether that's a problem. We're still looking further out beyond 40 days as the pandemic continues, but we would like to see it stay up a bit higher, but we don't know whether that's meaningful or not. Next slide, please. And I just want to show you some of the understanding the dissecting the immune response. These are two patients of some work that we did in collaboration with Weill Cornell, and breaking down the immune response to the IgG, IgM, and IgA. And you can see that it's interesting that the IgM response and the IgG response in our hands are actually coincident. You don't see much of an IgM response before the IgG appears, and there is certainly IgA. We're also looking in saliva as a antibody source, and I think that will be very, very interesting to monitor. So it's really important in individuals as well as populations to understand the immune response. Next slide, please. So the blood spot work is actually being used to do seroprevalence studies in populations around the state. And I'm sure these are data that I'm sure you've seen before on Governor Cuomo's daily addresses to the state. The highest seroprevalence is in New York City, which is not surprising. In grocery store random collections, we've done about nearly 16,000 at the moment, and the prevalence is as high as 19% in New York City areas, and around 14% a little further upstate. We've also looked in first responders, the fire department and the police department in New York, and the seroprevalence there is 17 in the fire department and 10.5 in the police department. Transit workers around 14.2%, and healthcare workers, I'm sorry, we've got a box covering this up. Sorry, 12%. So this is remember that this is a snapshot in time, and clearly it's going to be incredibly important to do multiple studies like this over time to see how the seroprevalence changes. And so this is something that we're really ramping up to do in very high volume, and will be important for monitoring the response. Next slide, please. So you've clearly got the message from both Florian and Jen that serology is important for seroprevalence studies where it works well, and to give an individual evidence of prior infection. We certainly need more study for returning to work without PPE. And this is such an important question as some states open up, and the immune status. And the immune status, we're really not going to know, I think, till we have reinfection examples, and these are going to take much longer. Next slide, please. And you can see many hands to do this work, and we're very grateful for our collaborations and all the work that's been done. And that's the end of my presentation. Thank you.
Great. Thank you very, very much, Dr. Taylor. So now we're going to go to our last talk, and then we should have about 10 minutes or so for some questions. I just want to remind people that our seminar is, our webinar is set to run through 12:15. So I hope those of you who can stay on through 12:15. So Dr. Jessica Metcalfe is an assistant professor of ecology, evolutionary biology, and public affairs at the Woodrow Wilson School at Princeton University. And she's a demographer with broad interests in evolutionary ecology, infectious disease dynamics, and public policy. She completed her PhD in ecology and evolutionary biology at the Imperial College in London. Dr. Metcalfe.
Great, thanks so much. So we're going to take a step back and think about the larger population consequences in the context of serology. And as I'm sure everyone is very aware, we're living in a time of multiple pathogen emergence events, starting from SARS in 2003 through to MERS, Ebola, Zika, and now SARS-CoV-2. And although we know more and more about these pathogens from the incredible technical innovations of the sort that has been outlined so far in this webinar, through to the nuances of the epidemiology, I think it's fair to say that every emergence event so far has contained surprises. So for example, H1N1 didn't attack older individuals so much. Ebola it was very surprising that it spread so far and so fast. And for SARS-CoV-2, for example, the case burden seems to be low in children, which is rather unexpected. And what I'm going to do in this talk is I'm going to sort of take the angle of how serology can help us think about these population level processes across a number of themes. And so I'm going to talk about transmission. I'm going to talk about the longer term, and particularly the question of herd immunity. I'm going to run through how susceptibility could be considered immune dark matter, echoing themes that Jill, Florian had brought up before. And I'm going to talk about serology to meet future pandemics. So if we're on this webinar at all, it's probably because sometime in late 2019, a coronavirus spilled over into human populations in China. And to understand subsequent spread, epidemiologists use two key values. One is called R0, which is the number of new infections per infected individual in a completely susceptible population. And the other is the serial interval. And so this is the time period that separates one infection from the next one. So just as a thought experiment, if you imagine an initial case in a population, then if R0 is 2 and the serial interval is about a week, 1 week later you'll have 2 more cases, in another week you have 4, and so on. And so essentially with these sorts of parameters, what you expect is that cases should double approximately every 2 weeks. For this particular coronavirus, the R0 is thought to be somewhere between 2 to 3, and the serial interval might be slightly less than a week. And of course, the consequence of that is that we get exponential growth. One of the multiple challenges that serology can contribute to in this space is that cases are very likely to underreport infections. We've all heard about the challenges around testing in this country and also elsewhere. And that leaves us with the question of, how many people could still be infected? How many people are still vulnerable to this infection? Which is one of the things that serological surveys of the sorts that have been described could really help us better understand the situation we're in now. Of course, the epidemiological process I described to you is a bit of a simplification. On average, it's possible that one infected individual infects 2 others, but in actual fact, there might be considerable variation in that process. And epidemiologists might describe that as super spreading events and the prevalence of super spreading events. The word overdispersion is often used as well. So if you imagine here we have 5 infected individuals at time 1, and then 1 time step later, one of them has infected 3, one of them has infected 2, and 2 of them have infected 1. And so you can see there's not too much variation here. 60% of cases at time 1 cause 80% of the cases a week later. And every single infected individual does cause some onward transmission. Now for SARS-CoV-2, we might be in a situation more like this one, where 1 individual infects 10 individuals, 3 individuals infect nobody, and 1 individual infects 2 individuals. So one way to characterize this is to say that 20% of individuals are causing 80% of the onward transmission. The consequences of this is that outbreaks are going to be more explosive. So when they take off really fast. but they also are more prone to extinction. So many introduction events might actually go nowhere, as shown in that second panel. We did some modeling early on in the outbreak to try and look at this. And based on introductions that had been observed at that time step, we found that the data suggested that maybe 10% of the cases were driving 80% of the onward transmission, which is quite a huge amount of variation. Again, serology might help us understand how and when such superspreading events might occur by giving us a much better window onto where the transmission is happening, especially for an infection that seems to be able to be asymptomatic as this one. So that's the early growth and the things that we might bring to bear to think about this. In the longer term, it's worth turning our heads to a different set of processes to think about how herd immunity might emerge. So I described susceptibles becoming infected in the early part of the outbreak. As you move into the longer time frame, you have to also consider the fact that once you've been infected, you're probably protected from reinfection for a while. And this is a theme that's also come up in previous talks. So just to illustrate this, again, I'm going to assume that I drop one infected individual, there, the little red dot, into a population of susceptible individuals shown by the black dots. And now I'm going to assume that R0 is 3. So if we drop this one infected individual in about a week later, perhaps, it will have infected 300 individuals. The proportion of the population that's infected will increase. The proportion of the population that is susceptible will also decline. And so as you move forwards, there's going to come a point where that infected individual will in some sense waste 2 of its infections. So 2 of the little arrows going out of it go nowhere because they are targeted at recovered individuals who are not at risk of becoming infected. And so if one infected individual is infecting less than one other individual, then the epidemic will turn over and the number of infected individuals will decline. And eventually you reach a point where if you drop one infected individual into the population, although there might be still a few susceptible individuals around, those are protected by the prevalence of these recovered individuals, these immune individuals. And this is referred to as herd immunity. This is a really useful piece of information because it tells us as well for infections for which we have vaccines, what the critical threshold for vaccination might be. So this is the proportion of the population that you need to vaccinate in order to ensure that the infection will be on a trajectory to extinction. So you can see that'll be where the infection curve turns over and goes from going up to starting going down. And there's some very simple maths that describes it. The proportion that you need to vaccinate is 1 minus 1 over R0, that magical value. This is, of course, an absolute cartoon of the real world. And in particular, even with an R0 of 3, the threshold that you need to reach to achieve herd immunity is probably slightly smaller because there's likely to be heterogeneities. These models assume that populations are well mixed, but in actual fact, we don't all have equal probabilities of encountering everyone else. But it's also worth noting that even once you've reached that point, that threshold for herd immunity, the downslope is long. So you're still going to be getting cases for an awfully long time as you move forwards. Clearly, having a sense, so serology will give us a sense of where we are across this landscape. And as Jill pointed out, multiple measurements are going to be really useful in understanding where we sit on this landscape. Okay, so susceptibility, so having described broadly the dynamics that undergo this emergence process and then what we might expect to be playing out over the slightly longer term course, we can turn to thinking about how serology in particular might illuminate these 2 processes. And Brian Grenfell and I often refer to susceptibility as immune dark matter. And so to think about this, I've shown, individuals, as I said on the previous slides, enter the world, probably start off susceptible, then enter the infected class at some rate, and then recover at some rate. Our window onto this process is usually just what we see from the infected box, so cases and deaths. And this, of course, is providing an incomplete window onto the epidemic and what's playing out. We're not seeing the susceptible box, we're not seeing the recovered box. But if we were able to pull together some serology by measuring antibodies, as described in the previous talks, we can complete this picture. So we can get a sense of the full trajectory and what might be occurring at that time. So to illustrate this, imagine that this is a real-time course of numbers of individuals infected as the weeks play through. In reality, of course, even though this is the true underlying process, we won't see this. So we might see, for example, 1 course reported out of 5. That may be quite optimistic in our current surveillance systems. We know that under the hood, there's also a proportion susceptible that will be falling as the number of infected individuals rise and then falling to some particular level. But early on in the outbreak, we will only have data, again, especially if we're not measuring susceptibility, we'll only have data from this sort of, first couple of weeks or so. And the challenge there is that you can fit lots of different models, lots of different flavors of the SIR model to this early time course. So for example, you might fit a model that had a particular reporting rate, say estimating reporting of 3 and 5 infections, that got transmission right, but assumed a very short duration of infection and perhaps assumed a later start to the outbreak. Or you might fit a sort of similarly good-fitting model, a good-looking model for that early phase, but that had much lower reporting, higher transmission, longer durations of infections, and an earlier start. So although those two things look very similar in the early parts of the case curve, you can see the difference play out as you go further forward through time. They look rather different in the susceptibility curve. So if we had data on those two curves from the corresponding to the blue dotted lines on the right-hand side, we'd have a much better sense of where we were in terms of the epidemiological trajectory. And this matters, of course, because it tells us, having a sense of what these core epidemiological parameters are, tell us how large this outbreak is likely to be, and what fraction of the population is likely to remain susceptible, and what the chances are that we've achieved that level of herd immunity. I don't think I need to say too much about this because the other speakers covered it so nicely, but of course we really need to understand what being seropositive means. And we're all hopeful that being seropositive means that you're not susceptible to reinfection. And this seems likely based on experimental evidence from similar coronaviruses, so not from this coronavirus, obviously, but from previously worked on coronaviruses. Some indirect mathematical models also suggest that previous coronaviruses show some immunity. And then the profile of immunity that we've seen following infection by this coronavirus looks very similar to what we might expect. And if we were able to establish this, and again, this is something that the others have said more eloquently, then we could start thinking about whether we could test healthcare workers or other essential professions and contribute to moving back to normality. But it's very much something that we don't know yet, but that would be extremely important to figure out and is one of the pieces that serology can help us grapple with. So I've sort of made the case that I think serology is a key piece in building mathematical models and understanding how transmission, how spread might happen, how many more individuals are left to infect. Has it been used and how can it be used to think about pandemics, this one, previous ones, future ones? So before thinking about pandemics, one might think about measles. So for example, prognostic serology, I think, has been used in interesting ways in thinking about measles dynamics. So in the 1990s, there was a cross-sectional annual age serology was used to trigger a vaccination campaign in England. A similar campaign was not triggered in Scotland, which experienced an outbreak. So this at least suggested that there was a useful forecasting ability in that serological data that told us, who was susceptible to infection and whether we were entering a dangerous zone. I was part of some work that predicted a high outbreak risk in Madagascar for measles based on a convenience sample of fever rash surveillance. So we published the work in 2018, and then very sadly, the outbreak occurred. So again, suggesting that despite the fact there hadn't been measles cases for a decade, the population was very much at risk. So what about pandemics? Well, I went looking around to see what I could find. In 2010, a retrospective on the H1N1 pandemic was published that suggested that representative serological surveys stood out as a critical source of data with which to reduce uncertainty around policy choices. I don't think it was widely deployed or made available, but the recognition has been there for a while that these things could really be game changers in terms of understanding the processes that we're interested in. And I think, there is such enormous potential to harness serology more broadly. There is, we have, everything from convenience samples of bloods to longitudinal age serology to really amplifying laboratory techniques as described. So I think it'd be an exciting time to start thinking about whether we could have a global immunological observatory as we do for so many other things. I know that we're running out of time, so I'll just say very quickly that there's so many things that from a broad epidemiological perspective serology can bring, from thinking about how many people are left to infect to understanding what seropositivity means for immunity, but also in mapping it to transmission models, we can get a grasp of aspects, mysteries such as what the role of children in transmission, if we also have additional data on contact patterns. And I think in understanding, situationally what the risk, because it gives us this window onto who has been infected, particularly in this context where so many infections may be inapparent, will be invaluable in better understanding burden and how it's distributed. So just a few resources, and that's all I have. Thank you.
Great. Thank you very, very much. That was terrific. We've had many questions come in, and we won't be able to cover all of them. I tried to just group a few of the questions together, and we'll direct them to our speakers. And I apologize to those who sent in very good questions that we're not going to be able to cover. So for either Jill or Jen, is the New York City lab or state labs, as far as you know, validating against a common gold standard? I have a couple of questions that I'm going to group together that are similar. Are labs sharing samples with each other to permit cross-validation? Can you tell us a little bit more about the prospects of saliva, which would be an easy specimen to collect for accurate serology? Why don't I pause there and let you tackle those questions? And then I have a few questions for Florian.
So this is Jill, and I will answer the question. Clearly, a gold standard panel of sera is important. It's something that we're working on with a number of the downstate hospitals, setting up a panel that we will fully characterize and share. And so the answer is yes, not yet, but coming. Saliva we're looking at, have not done a lot of work in yet, but clearly that and the fact that you can use saliva as a, clearly at different times during infection, this would be important. But yes, that could be a very good convenience sample. Jen, do you want to add to that?
Yeah, I will add, definitely labs share specimens all the time to do validations around the city and around the state. The other real advantage to being able to use saliva for a specimen for testing, either for an RNA-based test or for serology-based test, is that saliva is very easy to collect. People just need to spit in a tube, essentially. So it requires a lot less touch from a medical provider, which, as you all most likely know, there are shortages of PPE, shortages of collection devices, and being able to use these kind of alternative specimen types for all sorts of testing is really going to be key as we move forward and go into large scale testing across jurisdictions where a lot of people need to come in and get specimens collected. The less touch that requires, the more efficient we can make those collections.
Great, thank you. Okay, and then for Florian, and again, this is a grouping of a few questions. What do we know so far about the serologic response to SARS-CoV-2 among children and how that compares to adults? Any predictions, even if we don't have the data yet, from your perspective on what % of asymptomatics go on to develop detectable antibodies? Does the level of the antibody response indicate severity of illness or time since disease? And then the last one I'm going to put into this group of questions for you. If we're using serologic testing to identify convalescent plasma for clinical trials, doesn't that suggest that we think it's protective? So why can't we just go ahead and assume that these antibodies are protective?
All right, so the first three questions are relatively easy to answer. The last one is a speculation. So there is not widespread data for serology in children. That data is generated right now, but there have been reports of children getting infected where they were actually PCR confirmed and then they seroconverted. So we know that they can, and they probably do to a certain degree. Asymptomatics is also a good question. We have, I think, 2 or 3 asymptomatic people in our cohort that we caught because they were randomly PCR tested. and they made an antibody response, that does not mean that every asymptomatic but infected person does make one. That brings me to the next question. Yes, there is usually a correlation between the antibody response and the severity of disease. What we see is in a lot of hospitalized cases, the antibody response comes up very quickly and goes high. In the asymptomatics that we have in mild cases, it usually takes much longer to seroconvert. The kinetics seems to be different there. We see it coming up, but it usually comes later and might not go as high. Those are things that, you know, we need more data and to really kind of solidify these results, but that are the trends that we see here. Okay, the convalescent question. Of course, that's the assumption. The convalescent plasma therapy was used because there was nothing else, and people need to try something if there's nothing else. And it has been working well for other viral infections, like Hunin virus infections. It's been used for a couple of cases of measles, Hantaan infections, for example, and it has worked there, and in other cases it hasn't worked. Now, in terms of protection, there's things that we can extrapolate and kind of make common sense assumptions, right? We see that antibodies, and specifically neutralizing antibodies against common cold coronaviruses, protect for a certain time. It's very likely that we'll see protection from reinfection. Also, I don't think we have any kind of indication that any of the people who we tested positive here came back with a second infection. I mean, it's a short amount of time, but you know, there are indications. But on the other side, I think we have to be responsible, and as long as we don't have really the study that shows you are not getting reinfected for that amount of time, and if you have that titer, it would be irresponsible to say, oh, if somebody tests positive, you know, they can just go back to work without PPE, because that might not be a good idea, and people might engage in risky behavior. So, you know, I think a lot of people, a lot of scientists are very positive about the immune responses that we see, but we would also like to see solid proof that somebody who has antibodies and a certain level of antibodies is then really protected, right? I don't want to go around and say people are protected if they're not. So yes, we can assume, but we also need proof for that.
Great. Okay, well, I think just to respect everybody's time, we're now at 12:15. And again, we've had really excellent talks, and some fantastic questions have come in as well. But I think that we all have had an opportunity to learn quite a bit more about serologic testing. And we also are left with a lot of unanswered questions, but hopefully with more time, and as we all accumulate more experience, we'll be able to answer many of those questions. So I want to thank the speakers, and I want to thank also Treatment Action Group for helping to co-sponsor this webinar. The webinar has been recorded, and it will be posted on the ICAP website for those of you who either want to hear it again or pass it on to those who were not able to attend. So I think that's it. Thank you very much.